So my name's John. I work for a company called Driver Group. Right now we're in the process of working with hospitals, starting with UCSF, to process their late-stage lung cancer patients, process their tumor samples, figure out what it is that makes their tumor tick, figure out what it is that's driving their tumor and causing their cancer, and then provide personalized therapies for them. And then when we identify situations where there isn't a good personalized therapy for a specific group of cancer patients, identify the best drugs possible and actually run early stage clinical trials. So we're trying to cover quite a bit of the typical, going from basically early research all the way through drug development space. But today what I'm going to tell you guys about is first off a high-level overview of cancer, what causes it, what the molecular basis of it and what that means. And then I'm going to talk about how we actually turn tumors into text and what that text looks like and what that text means. And then the different types of information that we extract from this text. And then finally I'll get into a couple examples of how the field has turned this kind of text data into actionable clinical information. So first off, cancer is a disease of your normal cells. So it's a problem when one of the normal cells in your body, like a lung cell, for example, starts to divide uncontrollably and not act like a lung cell anymore, right? So it's a really interesting problem in that it's actually your own body, your own part of your own body that's just misbehaving and going uncontrollable. Luckily, a lot of different things have to go wrong in a cell before it'll actually turn into a tumor. So, there are lots of different safeguards. For example, your immune system is sitting there just waiting for a bunch of different mutations to pop up in one of your cells. And when it identifies a cell that has a lot of mutations, it'll attack that cell and kill it. That's what happens normally. Tumors figure out how to get around that process. Similarly, resisting cell death. So there are other mechanisms within a cell that cause a cell to commit suicide when there are too many mutations within that cell and it's just too messed up internally. So tumors figure out how to evade that. So those are just two of the examples of things that have to go wrong. And within any one of these, there are a lot of different ways that specific thing can get broken. So it's a pretty there are a lot of different things that you could be looking for. It's not a very simple world. So just to reiterate, it's not going to be one mutation, generally speaking, that goes wrong within a tumor. It's going to be an accumulation of mutations that slowly take a cell from a normal state where it's behaving properly all the way through potentially having uncontrolled growth. And just to clarify, when I say a mutation, what I mean is it's a change in your DNA, in your genome. So your genome is, it's like the blueprint for every cell in your body, what makes you you. So for example, you might have a couple extra letters, a couple extra ACTs and Gs inserted into a portion of your genome. And if that causes an important gene to break, that's not good. Okay, so the DNA of your genome, that's huge. That's 3 billion base pairs. The parts of that actually get turned into genes is about 1%. We call that the exome. So basically, there's this standard flow between DNA of the genome. You can focus in on just genes that get turned into RNA, which gets turned into protein. Proteins are the things that actually do stuff inside of your body. So the proteins are, they carry signals between your cells. They make up the structure of your cells. They make the cells do different things. So when you have a cancer, what's actually happening is you're getting mutations in your proteins or the part of the genome that regulates how much of those proteins are present. And that causes things like, for example here, perhaps this mutation in gene C is a little bit messed up. Maybe that caused gene A to turn off when it should have been on, and it might have caused gene B to turn on when it should have been off. So you can get these kind of signaling cascades that happen inside of tumors, these kind of like networks of crosstalk between different different genes that get mutated in different ways and signaling other genes that can cause a cell, a normal cell, to start misbehaving. So DNA is huge again. If you took the DNA in one cell, even though it's only about 10 atoms wide, a strand of DNA, only about 10 atoms wide, but if you unraveled all the DNA in just one cell of your body, and again, cells are so small you can't even see them unless you're looking at it under a microscope. If you unraveled that, it would be 1.5 meters long. So we're talking about a huge amount of information just crammed into a very small package. So how do we turn this into text? Well, I told you we do a normal versus tumor comparison. So we actually sequence both. We're looking for differences between a person's normal genome and their tumor genome. We're not interested in the things that just make a person unique as a person. We're interested in the things that make their tumor unique as a tumor. So we extract specifically the exome, which again is the part of the genome, the 1% of the genome that actually gets turned into proteins. And we extract the RNA, and we throw this into a sequencer. So this is about 55 million ACGs and Ts that we extract for one copy of the exome, but we have to sequence it many, many times over so that we're really confident in our observations, and for a couple other reasons I'll get to in a minute. So how much data is this? Well, if you imagine War and Peace, super huge book, about 1,400 pages of text, 3 million letters. We sequence about 6,000 copies worth of that for one person. So this is a lot of data to go through, and because of a current, I guess like a current limitation in technology that I don't really see getting over anytime soon, we have to do the equivalent of taking those bookshelves filled with books and throw them into a paper shredder, more or less. So what we get is this just big pile of data without any context, which is a lot of fun to go through. So what are we looking for in this big pile of data? Well, there are about 100 to 1,000 new mutations that show up in a person's tumor sample, right? In the DNA itself, you can get slight shifts in how much of the DNA is present, so you can get like extra chromosomes or parts of the chromosomes that are lost. So we have to identify those situations. Those can be really interesting too. And then also the RNA, which is a surrogate for how much protein is present, could be there at different amounts. So we have to pull all that out of this text data. So what does this look like? Let's zoom into one of these little fragments in this big pile here. It looks like this. You've got a little unique identifier that just tells you what that sequence is and where it existed in the imaging software that found it. The DNA sequence, the A's, C's, G's, and T's, and then individual confidence scores on each one of those letters, because the sequencer itself has a little bit of knowledge about how confident it is in every single base pair that it reads. So you can use that when you're trying to figure out what the data means and how much you believe things. So this is the first kind of thing that we look for, mutations. So what I'm showing here, one of those little gray bars is actually a sequencing read, one of these that I just showed you, aligned to the genome. So we give it context. We figure out where it most likely goes. It's kind of like a Google search, for example, where you just figure out like what it matches up with the most. So you do that for the normal sample and the tumor sample against a standard human reference. And then you can compare the two at every position in the alignment. And you can identify situations like this, where a certain set of the sequencing reads, a certain set of the DNA in the tumor, has a difference that isn't there in the normal sample. So for example, this right here is showing you a chunk of DNA that was actually deleted in this person's tumor. And this specific one is pretty important. If you have that specific mutation in your tumor, you're eligible for a drug called erlotinib, which extends your life pretty substantially. So these things are really important, and we have algorithms that identify them. I mentioned copy number alterations. This is the second thing in DNA that we look for. So this is when, for example, you have in one of your chromosomes, a chunk of that chromosome is just duplicated. It's there twice instead of once. So how do we find that? Well, actually, we look back at that alignment between the normal and the tumor. And in bins, we just count how many reads are there in the tumor versus the normal. And you get a ratio for that bin. And what you can do is you can plot out these ratios across a chromosome, and you can identify stretches where a whole bunch of sequential bins have a shifted mean of their ratios. And this is a pretty strong indicator that this person has an amplification, an extra copy of DNA at that position. So there are a lot of drugs that you can actually recommend just based on those kind of changes. And finally, RNA. This one's tricky. So instead of now showing you an alignment to a specific genome segment, what I'm showing you is an alignment to a specific gene. So you would count how many reads of the RNA are matching up with a certain gene in the tumor versus the normal. And you can plot this out, sort of an XY plot of normal versus the cancer sample. And there's a lot of noise in this kind of data, unfortunately. You've got about 20,000 genes. There's a lot of dispersion. A lot of different genes are just going to be like randomly overexpressed or underexpressed. It's really complicated stuff, but every once in a while you'll find really high magnitude outliers that you can really hang your hat on. And those are the situations that we look for. And also high-level patterns. So different tumor types will actually have different sets of genes that tumor type uses to basically to be a tumor. And that might be different from lung cancer and breast cancer, for example. So we've actually used that to categorize tumors of unknown origin, where the doctors had no idea where the tumor came from because it had already metastasized throughout the patient's body. So these tumors were everywhere, but we were able to look at the RNA in that person's tumor and compare it with this huge public resource and figure out, oh, this actually looks the most like a cervical carcinoma, for example. So there are a lot of really powerful things you can do with this. So now I'm going to shift gears and talk about a case study. So this is something that happened about, So this is something that happened about 5 years ago. There was this drug, I mentioned it a little while ago, erlotinib, that in a very small set of patients, when you give it to all lung cancer patients, in a very small set, about 10%, it really increased their survival. So in that subset of patients, they had really, really great survival. But when you averaged it across all patients, it didn't actually look like a very promising drug. So researchers were wondering whether there was a way to figure out who was going to benefit from this drug versus who wouldn't. Who should they just give regular chemotherapy and radiation to versus who they should definitely give this drug to. So what they also knew was that this drug targets a specific gene called EGFR. So they thought, well, maybe if we could figure out which patients had tumors that were dependent on the gene EGFR to be tumors, then maybe they could stratify the patients better. So here's what EGFR mutations look like in lung cancer. They affect about 8% of Europeans. It's actually different with different racial backgrounds, interestingly. And the other 92% of lung cancer patients are completely normal in this gene. So when they did the stratification, when they took just the patients that had a mutated EGFR versus the ones that didn't, 81% response instead of 8%. So this was definitely a clear way to stratify these patients. So this is actually the current FDA-approved test. If you have lung cancer, you go to the doctor, they'll do 2 tests. One is for this gene and one's for another gene called ALK to figure out if you're a candidate for either of these drugs versus just chemotherapy. All right, so what about right now? What about current problems in the field? So here's one. This is actually a really exciting new area. It's called immunotherapy. Earlier I mentioned that the immune system and evading the immune system was one thing that you had to do to basically be a successful tumor. So drugs that target the immune system are actually super effective in about 20% of lung cancer patients. The rest of them, not so much. However, when people have really drilled down on the single genes that have been involved in that these drugs are targeting, they haven't really found any correlates yet. So if you just look at that one gene, there aren't any correlates. So what I'm guessing is that this is going to be a much more complicated text mining problem, data science problem, where it's going to be a more interesting interaction between many different genes potentially that give you the signal for whether or not this tumor is going to respond. There's some indication that maybe just the mutation rate, how many differences there are in the person's tumor, is an indicator of whether or not they'll respond to this drug. But it's still very actively researched, and there's a lot of debate, and no one really knows. So if you want to get involved, that's a cool area. So anyways, text mining, the kind of stuff that you use to make recommendations on web pages and all that other stuff, you could also make recommendations for curing cancer. I'd like to think that's cooler. So where do you go from here? Let's say you're interested in this and you want to get involved. You could go to websites like rosalind.info. They have a lot of bioinformatics resources. You can learn about the field and the different algorithms and all that. You can contribute to open source projects like the amazing one Frank in this room is working on. Go to bdgenomics.org or listen to his talk next. And also, potentially, you could come work for me at Driver Group. So we're looking for DevOps people, sysadmins, software engineers, data scientists. So come talk to me afterwards if you are curious or interested. And now I'll open it up to questions.
Is the hypothesis that the difference is the only thing that matters a robust and tested hypothesis? Or is there like potentially unexplored linkages to the underlying genetics of the person to begin with?
Both, definitely both, right? Because, you know, I think one important clue is that, for example, in different racial groups, you get different levels of EGFR mutations. That suggests that there are like background differences in just people's DNA that, that correlates with whether or not they're likely to have a specific kind of lung cancer. There are also things like familial tumors that are, you know, you inherit certain mutations that make it more or less likely that you develop cancer by a certain young age. Or there's BRCA mutations. You've probably heard of that. They do BRCA tests now for a lot of people to figure out whether or not you know, you're likely to develop breast cancer. If you have a mutation in that gene, you're, I think, like 20x more likely or something, even though it's still a small number. Yeah. So, yes.
I was recently at a presentation by the CEO of MacMorks and Health, and they have this approach where they're using supercomputing to analyze the entire human genome, and then try to look at the cancer cells and try to figure out, you know, if there's a protein sequence of some sort that's out that they can then target very specifically, rather than just blast everything using chemo or something like that. How is, you know, what's your opinion on their approach and what they're trying to do, and how are you guys different?
I'm not super familiar with their approach. I think the big difference, there are other companies like Foundation Medicine that are also providing tests like this. I don't know how much of the genome NAND Health covers. We do cover the entire exome instead of just a targeted panel of known tumor-related genes because we're interested in research and discovering new things. But I think the biggest differentiator is the fact that we're also going after early-stage clinical trials. So this service that we're offering actually for free to patients, this isn't our business model. These reports that we're offering to patients for free is basically our Gmail, if you want to think about it that way. It's how we're getting data that we then use internally to make better decisions about which drugs to go after. Yeah. Yes?
How much of your work is, I guess, like compute or technique bound versus limited by actual real world data?
I'd say most of it right now is data limited. And I think one of the most challenging things is finding data sets that are well annotated. So where you know, for example, that a person responded a certain way to a certain drug. And that's part of why we're really focusing on collecting these data sets along with as rich response information as possible.
So how do you really do that? Do you focus, say, you go to a hospital and collect all the data, or you ask people around the country for them to crowdsource it, or?
So right now we're focusing on partnerships with specific hospitals. So we're starting with UC San Francisco, and we're working on expanding that to the entire UC system. I think we're in some talks now with some hospitals in China as well. So yeah. Yes?
Do you have another example for the relationship between text analytics and your sequencing?
I don't have other examples right now. One of the things that's a commonality is the stacks that we end up using. So for example, Frank in a little bit is going to talk about his tool, which we're working on incorporating into our own pipeline, which uses Spark and a couple of other things like that. But yeah, I mean, really it's all, we turn these tumors into text data. And then at that point, you know, there are a lot of the same kinds of algorithms that you apply to the machine learning problems with this kind of text data versus other kinds of text data.
Text data is just a sequence of images.
Yeah.
So usually the example is a single gene or single application. So if you do have, and you also give a counterexample to say that example is not due to a single mutation or something. So in that second case, what is the general approach you would take to tackle the problem?
Yeah, so I would say it's a tough question, you know, because there are a lot of people working on this right now. I would start looking at a lot of different features and develop hypotheses about, you know, these different features and then try them out. So one of the things that we're thinking about doing internally, right now a lot of people are just using the raw counts of mutations, but mutations can appear in tumors at different frequencies, for example. So for example, maybe every single cell in one of your tumors has a mutation versus maybe only 10% of your cells in your tumor have a particular mutation versus, you know, the other 10% have another mutation, another 10% have another one, and so on. So you can get different levels of what's called heterogeneity inside of your tumor. So it might be that even tumors with a large number of mutations, if they are heterogeneous versus not heterogeneous, they might actually respond differently. And you can imagine that maybe the immune system could attack a tumor cell better, and you have to think back to like what it is that the immune system's recognizing. It's recognizing mutated proteins. So it might be able to attack and learn how to kill a tumor better if all the tumors are showing the same set of mutated proteins versus a whole bunch of different kinds of mutated proteins, right? So that's one of the things that we're working on right now are pretty advanced normalization methods that can answer those kinds of questions, whether or not mutations are mostly at high frequency versus low frequency. All right, thank you.